As always, we'll start by looking at our warm-up. All right, so gonna share screen. And yeah, this, in case you're wondering what this is, my friend has one of these. They're these crazy, like, automatic self-cleaning. After the cat like pees in there, it automatically goes kind of gets, yeah, it's just like a self-cleaning kitty litter thing. Their cat seems to be okay. I think my cats would just freak out. Okay. So this was, I was very happy to see that everyone that I graded here, people really did get that the kidneys and the urinary system are doing a lot of important stuff beyond just excreting wastes. So yes, they excrete waste, particularly like urea and nitrogenous waste and drug metabolites, but then they're also critical for like water balance, electrolyte balance, pH balance, and a variety of other things. So, and that's going to be something we'll be, kind of really focusing in on the lab today as well. Tubes that carry urine from the kidneys to the bladder are the ureters, and everybody got that as well. Almost everyone got this. The filtration is what's going on in the glomerulus. Filtration, that's where you have this kind of indiscriminate leakage of water and small solutes out of the blood into the tubule. And again, large volumes, like I said, like 180 liters per day of filtrate being formed. And then the majority of that is getting reabsorbed back into the body as it goes through the tubule. And we're going to look at that in much more detail in just a few moments. And there's also secretion. Tubular secretion is also going on, where there's still stuff in the blood that you haven't actually got rid of that you're going to move from the blood into the tubule, so it will eventually leave your body as urine or part of your urine. Choose all that are true concerning the glomerulus. Definitely not found in the bladder. It's up in the kidneys. And it's a capillary bed fed and drained by arterioles, because it's actually one of these portal system things. The efferent arteriole is going to feed into the peritubular capillaries, which is where you're going to be reabsorbing, all of the water and solutes that are part of the filtrate that you want to keep. It is not actively pumping solutes across its walls. Things are it's this passive filtration. It's just that we talked about that net filtration pressure. The pressure within the little capillary is just kind of forcing the leakage out through the fenestrated capillaries. So everybody got, though, it's made of very leaky capillaries. Everybody got this too. If your mean arterial blood pressure is too low, glomerular filtration may slow down or even stop. And that's like a danger of really low blood pressure, is renal failure, and also why your kidneys are going to be an important part of keeping your blood pressure up, because you want to make sure that you have adequate blood pressure to maintain filtration. Although we'll talk a little bit, like parasympathetic response actually kind of restricts the afferent arterioles and kind of limits, temporarily blood flow into the kidneys, because you want the blood going off to your muscles and this or that. You don't want such a huge proportion of your cardiac output going into your kidneys if you need it in the moment to, get away from the tiger or whatever. All right. So are there any kind of questions about any of the parts of the urinary system we've covered up to this point. All right, so what we're going to do now is we're going to get back into the kidneys, back into the nephron. I kind of quickly introduced a lot of the ideas of PCT and loop, Henle, and this and that, but we're going to slow down and look at each of those and kind of see what's happening in there and how kind of the adjustment of the filtrate and the ultimate composition of the urine happens. All right, so I'm going to go look at I'm going to share the PowerPoint because it has some of the nice pictures, I think, just to kind of, so we kind of get situated what we're talking about here. Again, remember the idea. Let me get my little kidneys are here. This is where the action is happening. This is where all that processing, and again, they're getting like 20 to 25% of your cardiac output and processing. Here's the kidney. Remember that there is the cortex, the renal cortex, this kind of outer part. This is where the nephrons start. That's where the little glomeruli are. And this cortex is also kind of your normal osmolarity. It's 300 milliosmolar. We're going to be following osmolarity a lot in this kind of discussion of the nephron. The osmolarity both of the filtrate at different points along the tubule, as well as the osmolarity in the interstitium, and just outside the tubule, just in the space between the cells, because that's going to be determining whether things get sucked in or out of the tubule. So the medulla, I can change the color of this little dude here. The medulla is this inner part here. I can't really read it anymore now that I've made it too dark. But like I said, there's this gradient. Like as you go from this, 300 in the 300 milliosmolar in the cortex, as you get into the deepest parts of the medulla, it gets all the way down to maybe like 1200 milliosmolar, 4 times as high concentration. And sometimes I'll be loosey-goosey, I'll just say like way saltier, but I don't really mean just salt, just all solutes, what I mean. But it's just easy to say it's 4 times saltier deep in the medulla instead of saying it's 4 times the osmolarity dissolved particles per liter, moles of particles per liter. All right, so what we're going to do is follow the nephron. And let me put my ink back to a color that you can actually see through. So here we talked about the major arteries coming in, then they branch and ultimately feed like 1 million little afferent arterioles feeding into 1 million little glomeruli, and each glomerulus being the beginning of this nephron that is going to be the thing we're going to talk about now, the structural and functional unit in the kidney. Some of the nephrons are kind of more in the cortex. In fact, the majority in the human are up here. They call them cortical nephrons. But other ones, maybe about a 15%, actually dip very deep into the medulla. They're called juxtamedullary nephrons. Those are going to be really important, particularly for creating this osmotic gradient as you go down into the medulla. So we'll definitely be talking about these juxtamedullary nephrons as well. And again, this is kind of a picture again. This would be like a cortical nephron, a juxtamedullary nephron that's going down deeper. But in both cases, you've got the glomerulus that feeds into this proximal convoluted tubule, this loop of Henle, a distal convoluted tubule then they feed into these things called the collecting ducts. Collecting duct. And that collecting duct is going to be the final place of processing of the filtrate before it becomes urine. After you leave the collecting duct, that's what we would just call urine, or I'll write it out P. It's less I have to try to do with my mouse here. And then remember that the nephrons are always in this intimate association with the peritubular capillaries. Again, what are the two processes that are, so we have filtration happening in the glomerulus to create the filtrate, but what processes are going to be happening between the tubule and the peritubular capillaries for the rest of the journey?
Secretion and reabsorption.
Yeah, exactly. So there's that tubular secretion and tubular reabsorption. Tubular reabsorption, where you're pulling stuff back into the body from the that's inside the tubule. Secretion is if there's something that's still in the tubule, still in the body you want to get rid of, you can move it into the tubule so it'll eventually leave your body in your urine. All right. So this is what we're going to talk about now, and I'm going to be drawing. But again, we're going to start at the glomerulus where you're making the filtrate. Then we'll look at what's going on in the proximal convoluted tubule. We'll look at the loop of Henle, which has both a descending limb and an ascending limb, which are very different from each other. Together, the descending and ascending limb of the loop of Henle are what's going to create that osmotic gradient. Like I said, in the cortex, it's about 300 milliosmolar. Once you get down into these deeper parts, where the tip of the loops of Henle down are, you're up to like 1200 milliosmolar. So we're going to see how that gradient is created by the loop of Henle. Distal convoluted tubule, we'll be doing some stuff. And finally, the collecting duct. Again, the collecting duct is going to be where we finally meet ADH, antidiuretic hormone again, and aldosterone. We talked more generically about what they do, but in our discussion today, we'll talk more explicitly like, oh, they are doing these specific things, turning on these channels in the collecting duct, causing reabsorption of water or like salts. So let's do this. All right, and I will, there is a lot going on. They don't call them the convoluted tubules for nothing. So you have to kind of just follow along. And at each stage, we're going to be following, again, both kind of the amount of filtrate, the volume, how much has been created, how much has been reabsorbed, as well as the osmolarity, because that's going to be important as well. So let's start with the glomerulus. So I have my afferent arteriole. Feeding in my glomerulus. And like I said, the glomerulus is leaky capillaries under high pressure. So this is where the filtrate is getting formed. And we're going to catch it in this renal corpuscle or Bowman's capsule. So this is the beginning of the whole process. Making the filtrate here. And if I, and also I should, let me put in a, right, just remember we're at like 300 milliosmolar. That's bad. $300 milliosmolar here. Again, as we get further down into the medulla, it's going to get as high as 1200 milliosmolar. So this is, we're up in the cortex here. This is medulla. So if we look at the filtrate that's being formed here, we've already talked about how much is getting made. We said that, maybe I'll go onto another screen. You know, filtrate is around 10% of the kind of incoming blood flow. And we know that, you know, kidneys, let's make life easy. Let's say get about 20% of cardiac output. So normal cardiac output is about 5 liters. What am I saying here? 5 liters per minute. So that means the kidneys are getting around 1 liter per minute, right? In reality, it's like 1 to 1.25, but to make the math easier for the rest of our discussion, let's just say 1 liter per minute. Just makes our life easy. Which means, in terms of filtrate production, we're getting about 100 mils per minute. So this is the number that we're going to start with. If you look across, you know, the million nephrons in each kidney and just look at all of the filtrate that's being formed, every minute there's about 100 milliliters of filtrate that is being produced in your kidneys. So that is going to be our starting point for our discussion of what's going on in the nephron. I'm going to draw one nephron, but just imagine it happening a million times in parallel in each kidney. So does this make sense? Are there any questions about how I get there's 100 mils per minute of this filtrate being formed? Right, okay, so then if we look here, it's about 100 mils per minute is the amount of filtrate that's being formed. What is the osmolarity of that filtrate? You know, and I can give you a major hint. You're leaking out indiscriminately water and solutes together, kind of in equal proportions. So what would you expect the osmolarity to be of that filtrate?
Around 300 milliosmolar?
Yeah, exactly. It's going to be the normal osmolarity of body fluids. So this is the beginning of this whole process. You know, again, we know things are going to change dramatically. One, because if we don't reabsorb most of this volume, we would be peeing out 180 liters of pee, which we don't do. Also, we know that urine can be either really dilute, almost like water, or it can be really super concentrated. So we're going to have a lot of opportunity to change the concentration, the osmolarity of the what's ultimately going to become the urine. Remember, and that's that the specific gravity is basically a measure of that osmolarity, how much dissolved stuff is in there. Higher specific gravity means higher osmolarity. Lower specific gravity, when you get to 1, it's basically saying it's just like water. All right. So the first step is going to be the PCT, the proximal convoluted tubule. I'll draw it all kind of twisty. Oh, and I can use my little typer to make this easier. So the proximal convoluted tubule, this is going to be primarily where there's a lot of reabsorption happening. So like 65% of the water. 60, and again, we have the peritubular capillaries coming in here, right? We have that efferent arteriole, which then feeds into the peritubular capillaries. The peritubular capillaries is where the water is coming back in, right? The water is being moved from in the tubule back into the bloodstream. So, and not just water, we're also absorbing a lot of solutes. This is like, all the glucose is getting reabsorbed here in the PCT, as well as lots of other stuff. Remember we talked about in diabetes is if there's so much glucose that you can't reabsorb it all by the time you get past the PCT. But normally during the passage of this filtrate through, it's moving down here, zooping around in this PCT, by the time it's made it through the PCT, all the glucose has been returned into the bloodstream and it's not going to show up in your urine. So by the time, and there is some secretion, like maybe I should, just for completeness, I will put that here as well. So lots of reabsorption, but also some secretion. Like for instance, like protons. Like if you are trying to adjust your pH balance and things are too acidic and you have to eventually pee out some protons, H pluses. This is one of the places where you can have H pluses moved from the blood into the tubule so they eventually leave your body to help adjust your pH balance. But when I think of PCT, I usually think of it mainly as absorption. In anatomy, some of you probably remember, if you look in the lumen of the tubule here, it's got all these microvilli for increased surface area for absorption. They're really fuzzy looking lumens. So once we're done there, we're going to get to the loop of Henle. So, but before we really talk about the loop of Henle, let's check in with our filtrate. So we were saying when we first looked at this filtrate being formed, the kind of volume is about 100 mils per minute, and the osmolarity is 300 milliosmolar, the same. So let's check in with what does the filtrate look like here after we've just finished going through the PCT right before we enter into the loop of Henle. So if we have reabsorbed 65% of the H2O, how much does that leave left? Can't hear anybody. What's 100 minus 65? Well, say it again.
35?
Yeah, yeah. I kind of want to just make sure people are staying awake here. So maybe 35 mils per minute left at this point. So we've already reabsorbed, 2/3 of the water, but we're still going to have to absorb more still, because at this point you're still going to have like 40 liters of pee if you don't pull back more. So there's 35 mils per minute now of kind of volume. And in terms of osmolarity, it's going to be pretty much the same though, because we're reabsorbing water and solutes. So we're reabsorbing water and solutes, so we're still at 300 milliosmolar as we enter into the loop of Henle. So the loop of Henle, which it wasn't the dude from the Eagles. It was some other anatomist a long time ago. Kind of first described this thing. The two limbs, we have the descending limb. Let me draw this better. And the ascending limb. In general, the descending limb is squamous cells. The ascending limb are thicker cells, but it doesn't matter for you guys. What is going to be important, and I'm going to actually erase some of this other stuff to give me more room here. Yeah, let me just PCT reabsorption, H2O and solutes. So the descending limb of the loop of Henle has aquaporins. So the word aquaporin it just means a water pore. We talked about, previously, like water tends to be able to make it through membranes. That's why osmosis is a thing. However, when you have specialized places where you really want to control whether water moves across or not, you can put a lot more cholesterol in the membrane, make the little like phospholipid tails much more tightly packed and stuff. And then you can actually control. So we can have cells where H2O cannot cross on its own. Ooh, it can't get across. But if you put in an aquaporin, then water can cross. So we're going to use these in the kidney tubules some places are just going to be always permeable to water or not permeable to water. And when we get to the collecting duct, we're actually going to be able to adjust the numbers of aquaporins and actually change the permeability to water in order to absorb more or less water, depending on whether you need dilute or concentrated urine. So aquaporin is just a fancy word for a little membrane protein that acts as a water pore that allows water to go through these membranes. So this descending limb is permeable to water. Not solute. So maybe I should put a little yellow highlighter around it or something. What? Yellow. So defining qualities of the descending limb: permeable to water, not to solute. And like I said, this is going to get kind of confusing because each part of the tubule are going to have very different properties, but they'll all kind of work together. Ascending limb is going to be really different. Ascending limb is not permeable to water. But actively pumps salt out of the tubule back into the body. So all right, so let's talk about what this means now and what happens here. This is it's cool, but it gets a little complicated. So we're going to let's start with the assumption that the medulla is really salty. It's like 1200. So if we look just at the it's 1200 milliosmolar outside here. Ready? It started at 300 milliosmolar when we're up near the cortex, but as we go down towards the deeper parts of the medulla, it's becoming like 1200 milliosmolar here. So that's kind of in this interstitium, in the places outside the tubule here. So imagine now what's going to happen. We have when we last met our filtrate, let me make a bigger one here. We had our filtrate here. We had 35 mils per minute coming through, 300 milliosmolar, and now it is going to continue moving down the loop of Henle. What is going to happen if you have something that's 300 milliosmolar starting to dive down into this region where it's 1200 milliosmolar and the walls are permeable to water?
A lot of water is gonna leave.
Yeah, exactly. This is the classic setup for osmosis, right? Semi-permeable. The solute is not moving, but water can move, and it's way saltier outside than inside. The water is going to suck out of the tube into the space, into the space outside of the tube. So water is going to come out. Maybe I'll do black. Be easier to see. That wasn't black. So H2O is leaving this descending limb. So that means by the time we get to the very bottom, like let me get rid of this. If we keep talking about what does the filtrate look like at each of these stages. If we take a look right here, we will have reabsorbed another 20% of the original volume. So we start entering here around 35 mils per minute. By the time we are down there, we're down to like 15 mils per minute. Right, so at this point, we have reabsorbed like 85% of the original volume that started at the beginning. So we've reabsorbed a whole lot of water, what is the osmolarity going to be like when we get here down to the bottom of the loop of Henle? After all that water has left the tubule, but the solutes are still trapped inside.
It's going to be much higher than it was before.
Yeah, it's going to be much higher. And in fact, it's going to basically equilibrate. The water is going to keep leaving until the inside and the outside no longer have a gradient, right? So by the time we get to the very bottom of the loop of Henle, the osmolarity is going to be like 1200 milliosmolar for our filtrate as well, right? If water can move, the water is going to keep moving until there's a balance. It's kind of like it's an osmometer, right? As long as there's a gradient, water will keep moving, and finally, when there's no more gradient, then there's no more movement of water. Like that's why we had we spent like we were beating a dead horse or whatever with osmolarity and stuff, because it's really at the core of so many things here. Kind of understanding a concentration gradient is what's driving things. Hopefully makes this feel more intuitive now. So now we are going to start rising up. And we actually had one of the I just heard this like a week ago. I'd never heard it, but somebody was saying one of the mnemonics that they use for trying to remember which side of the loop of Henle is permeable to water or not is like, kind of water falls downhill. So like the descending one is where the water is falling, leaving out of the tubule. So if that's useful, I don't know. But you want to keep track that the descending limb is where the water is leaving. Once we get to the ascending limb, not permeable to water and actively pumping out salt. And it can pump salt. Where's the power going to come to pump the salt?
You're going back up the osmotic gradient.
But if it's 1200 here, you actually pump the salt out till it's only like 1100, actually 1000 in here. Whatever you can pump can maintain gradient. Up to 200 milliosmolar difference. So actually up here, I can pump sodium out until it's 100 milliosmolar in the tubule, as opposed to 300 milliosmolar outside. So if the gradient becomes greater than 200, then it's pushing back too hard for my pump to push against. But I can basically pump sodium out. It's kind of like, let's say you had like you're in your yard and you get a fish fountain or something, which is basically, there's a pipe in the little fish sculpture. And then you actually have a pump here. And you've got water. Yeah, I'm being a little slower with some of my drawings because they changed my little interface here. I've got more options to do shapes and things, but my normal like clicking is like I'm landing on the wrong things. So I've got water. Come on, you silly goose. There it is. I have water here. The pump is pushing the water against gravity. I've got some height above the ground. How high this fountain is going to go depends on how strong the pump is, right? I can maintain a height fighting gravity of a certain height depending on how strong the pump is. These like sodium channels that are in the ascending limb of the loop of Henle, they can pump hard enough that whatever the gradient is on one side, they can go against the gradient to maintain a difference of up to 200 milliosmolar difference between the two sides. Once the gradient pushing back is greater than 200 milliosmolar, then there's not enough power left to keep pushing more sodium out, if that makes sense. So back to this crazy messy drawing here. Here in the ascending limb, I'm pumping sodium out. I can maintain a gradient up to 200 times 200 milliosmolar. So by the time I get to the top here, let me just give you, I guess I already did give it to you. I am at 100 milliosmolar. Let me erase this and make it prettier. It's 100 milliosmolar inside the tubule because, you know, when I'm down here, when it's like 1200 outside, it's like 1000 inside. You know, if I was at the level where it was 600 outside, it would be 400 inside. By the time I get up to here where it's 300 outside, it's 100 milliosmolar inside. This is going to be the way that we can have urine that is hypotonic to your body fluids, right? Your urine can be less osmotic than your normal 300 milliosmolar, right? If we don't, you know, this is how we're going to be able to have hypotonic urine, is because we're pumping this solute out of the tubule. So what's going to ultimately leaving the body is actually more dilute than your normal body fluids. You know, in terms of volume, because this is not permeable to water, you don't really change the volume. No water is leaving or coming in. So if it was 15 mils per minute down here, it's still going to be 15 mils per minute up here. So what I'm going to do now, I'm going to do this briefly. I don't want to get lost too much in this. But I do want to explain a little bit how this stuff, if I'm going to, at this point, I'm going to get rid of this in a moment so it can just get messier. The qualities of this loop of Henle, having this descending limb that is permeable to water but not solute, how having this ascending limb that pumps out sodium but is impermeable to water. If you just take this loop of Henle structure with these properties and let it go, it creates this gradient. So loop of Henle creates and maintains osmotic gradient in the medulla. I don't want to get lost in the details, but I will show you just a little bit of how it works because it's really cool. And this is something that it's not easy to do with me drawing. It's way easier to show as a little video. So it's a video I've linked into the module. I'm just going to show you, I'm not going to run through the entire thing, but I'll show you a little snippets of it so you can get a sense of how it works. So if we go down to, where is it? Here we go. I just have it here, YouTube supplement on how loop of Henle works.
Oh my gosh, I failed the USMLE again. I can't believe this. How many times am I?
Hiya, welcome to Biomed Sessions with me, Ruse. Today we're going to be discussing the loop of Henle.
All right, so Ruse is going to discuss the loop of Henle. It's worth going back and looking at this in more detail later, but I'm just going to kind of fast forward here a little bit. All right, so basically what she is saying is, let's do a thought experiment. Let's do a thought experiment where this is the loop of Henle right here. And at time equals 0, just assume everything is at 300 milliosmolar. There's no gradient to start with. The only thing we need to assume is that the descending limb, which is on the left here, is impermeable to solute but permeable to water. And the ascending limb, which is the thicker looking one here on the right, is not permeable to water but pumps out salt. And then, this is showing the osmolarity inside the tubule of the filtrate. This is showing the osmolarity outside the tubule, just in the interstitial fluid between the cells. And now what we're going to do is we're going to turn it on. We're going to turn the pumps on, and there's two things going on. There's the pumping of the sodium out of this tube. There's osmosis that can happen on this, but then there's the flow down the descending and up the ascending. And let's see what happens.
So in the latter part of the loop of Henle, our aim is to create a difference of 200 milliosmol per liter between the ascending limb and the interstitial fluid.
That's just going to be from ATP-powered sodium pumps, pushing sodium out of the tubule into the interstitium.
The only way to do this is by pumping out sodium ions. So let's pump. As you can see, the osmolarity in the ascending limb has decreased due to a loss of sodium, whereas in the interstitial fluid, it has increased due to a gain in sodium. And if you look closely at the values, you will see that we have achieved the 200 milliosmol per liter difference, otherwise known as a gradient. But what's happening with the descending limb? Here the filtrate needs to equilibrate, meaning that the water will leave passively until the filtrate in the descending limb reaches the same osmolarity as the interstitial fluid. Time to equilibrate. Note, equilibrating does not change the osmolarity of the interstitial fluid.
So this is just the first step. We pumped out sodium from the ascending limb until we have a 200 milliosmolar difference. And then the other side, though, is always going to be doing osmosis based on what the outside osmolarity is. So we're going to, and now what's going to happen, though, is there's the flow. The stuff that's in the descending limb is going to start going up the other side.
Filtrate moves through the nephron continuously, but I'm going to break this down into steps for you. Let's advance the fluid in the loop of Henle. When brand new 300 milliosmol per liter filtrate enters the descending limb from the proximal convoluted tubule, the filtrate already in the loop of Henle is pushed further along. Bringing back our values for the interstitial fluid, we see that our gradient has been messed up. So we need to re-establish that ideal 200 milliosmol per liter difference. Let's pump out some sodium ions. Okay, although the values vary, there is still a difference of 200 at all levels, so we're content. Now to equilibrate the descending limb with the interstitial fluid and advance the fluid in the loop of Henle a little. Lo and behold, the gradient is messed up again, but we know what to do. Let's pump and establish that 200 milliosmol per liter gradient. Equilibrate to match osmolarities and advance the fluid. Okay, one more time to get the point across. Our gradient is messed up. Pump out sodium to restore the gradient and equilibrate. Alright, so looking at our values for the interstitial fluid, we can see that the osmolarity increases the deeper you go into the medulla. As all that pumping and equilibrating continues, we eventually reach a maximum concentration of around 1,200 milliosmols per liter. The entire process that I have just described is called countercurrent multiplication. Countercurrent because the filtrate flows in opposite directions in the limbs of the loop of Henle, and multiplication because this countercurrent flow enables the effects of the gradient to be increased, i.e. multiplied. So in summary, the The loops of Henle of juxtamedullary nephrons are involved in the process of countercurrent multiplication, which enables the interstitial fluid to become more concentrated, i.e. increase in osmolarity the deeper you go into the medulla. The high concentrations in the medulla facilitate the passive movement of water out of the collecting duct and result in more concentrated urine. Okay, hopefully this tutorial...
All right, so let me stop sharing. So I think she does a really good job of making a really kind of interesting but kind of complicated thing like understandable. And that process, that countercurrent multiplication, there's a little more going on. There are specialized capillaries, the vasa recta, that go down into there that are carrying the water away, that make sure we don't destroy the gradient and stuff. And don't worry, you are not responsible for knowing the details of how the vasa recta are special and all that. But another reason to really understand that this is going on here is, we've talked so far about two different kinds of blood pressure meds. We've talked about the beta blockers, the beta adrenergic antagonists to lower cardiac output and lower your blood pressure. We've talked about how ACE inhibitors are going to block the production of angiotensin II, which is going to keep your blood pressure down. Another really common blood pressure med are these loop diuretics. Lasix. Most people have probably heard of Lasix or loop diuretics. Loop diuretic, diuretic, something that makes you pee more, is called a loop diuretic because it is working at the loop of Henle. These basically block the sodium pumps in the loop, the ascending limb of the loop of Henle. And so you can kind of imagine from that whole story we've just seen, if those sodium pumps are not doing their thing, what is that? How is that going to change the whole overall environment in the kidney? Right? Remember, a big part, let's pump. What happens what was the result of all that? Like, let's pump and re-equilibrate. But I actually can't say, I've actually hung out with a British guy this weekend, and I asked him, do you guys get annoyed if we like pretend using British accents? And he says, no, we find it kind of amusing. So I don't feel, I double checked. What's going to happen? Chris?
Will it lower the osmolarity in the medulla because we're not pumping out?
Exactly. You're going to have a lower the kind of osmotic gradient in the medulla. Right? The reason we got that 1200 milliosmolar was the action of those sodium pumps, pumping out the sodium and then having that countercurrent multiplication. But you're not going to have that working as well if your sodium pumps aren't doing their job as well. So things aren't going to be 1200 milliosmolar. There'll be less, which means if you have less of an osmotic gradient, you're not going to be pulling back as much water into the body. You're going to be peeing more. So loop diuretics work by lowering the osmolarity. I should say maybe lower osmolarity in the medulla makes more sense. Lowers the osmolarity in the medulla, which means you're not going to be pulling back as much water into your body, and more water will be leaving your body, less blood volume, less blood pressure. So again, another very common med prescribed to try to control hypertension are loop diuretics. Keep those sodium pumps from doing the countercurrent multiplication and increasing the osmolarity deep into the medulla. You will release more water. So does that make sense? Okay. So back to our nephron. We are almost done with this puppy here. And here we have our cortex. Here's the medulla. Again, like I said, it's somewhere around just interstitial around 300 milliosmolar when you're up around here. By the time you get into the deepest parts of the medulla, it's like 1200 milliosmolar. That's going to be important. Back to the nephron. Glomerulus. PCT, which we've just been talking about. Loop of Henle. Which we've just been talking about. The next part is going to be the distal convoluted tubule. I'm going to make it do a little weird deal here. So the distal convoluted tubule is a place where it's mainly secretion. Again, in anatomy, the way you can tell you're looking at a slice of the DCT versus the PCT is that the lumen is smooth. You don't have all those microvilli optimized for reabsorption. Right? If you look at a cross section of the DCT, it just looks like that. If you look at a cross section of the PCT, it looks totally furry inside with all these microvilli to reabsorb stuff. So what that means is we don't really change much around the volume or the osmolarity here. There's a few little things getting like dumped in, but in reality, if we look around here, it's going to still be about where we were at 15 mils per minute and around 100 milliosmolar. Right, that's kind of what we're entering in and what we're kind of leaving with. There is this little weirdness that I just talked about, or that I drew here. We are going to come back here. This place where the DCT kisses the afferent and efferent arterioles, that is the Juxtaglomerular Apparatus. That is the place where renin is going to be released and all that. So we'll come back to this place in a few moments. Maybe I can type it real quick just so you have words for it. Again, we'll come back there, but just so you kind of see where it is, it's that place where the DCT kind of comes back and touches and has specialized cells along with the arterioles. And then the last thing we need to talk about for urine formation is the collecting duct. So this dumps into this is my collecting duct. So the collecting duct is the final place where this stuff goes through what was filtrate, and by the time you make it out to the bottom of the collecting duct, you are now to the ureters, and you're going to leave the body. So this, whatever it comes out at the very end of the collecting duct, is actual urine. That's the stuff that you're going to pee out of your body. You have more than one nephron feeding into the collecting duct. You can have lots of nephrons feeding into the same collecting duct. So here we have our nephron doing everything it's doing. And at this place, this is where we can do kind of final adjustment of concentrations of water, of sodium. Like this is stuff, this is where the action is happening in our lab for later today. Depending on your body's needs, the properties of the collecting duct are going to change. So for instance, if ADH present, insert aquaporins in here. So if there's no ADH present, we've got 15 mils per minute coming here. It would just go through, because if there is no aquaporins, then it doesn't matter what's inside or outside. It's just going to continue down. You're going to have large amounts of very dilute urine, right? This is very dilute, and this is actually, again, 15 mils per minute. That's a huge amount of urine. If you do have ADH present, you insert aquaporins in here. And now, as the stuff is 100 milliosmolar in here, but it's like 1200 milliosmolar outside, the water is going to totally come back into the body. And what actually leaves the body as urine is going to be much less in volume, right? So ADH, antidiuretic, stops you from peeing. You don't pee as much because with aquaporins, the water comes back into your body rather than being part of your urine that leaves your body. So this is where ADH is doing its magic. And then we also talked about ADH having other roles. It's a vasoconstrictor. It's also talking to your hypothalamus to increase thirst. But in terms of telling the kidneys to not release as much water, this is how it does it. It inserts aquaporins into the collecting duct. The other hormone that was part of the renin-angiotensin mechanism that works here is aldosterone. Aldosterone turns on sodium pumps to reclaim more sodium from the tubule. So let's put that in here. turn on sodium pumps. Right, this is, the group that drinks just pure water and doesn't eat any salt of any kind. Their body's like, I'm kind of low on sodium. I don't want to pee out my sodium. I want to keep it in my body. You're going to have more aldosterone. And that aldosterone will turn on these sodium pumps here and keep it in your body. If you did eat a bunch of salt and your body is like, whoa, I got a lot of salt, I got to get rid of some of this to maintain salt balance, then you won't have aldosterone, you don't reclaim it, and then the sodium comes out in your pee. Right? That makes sense? So both of these, ADH or aldosterone, they'll be controlled by your body's needs to either conserve or let go of water, to conserve or let go of sodium. And as we saw in the renin-angiotensin mechanism, if you turn both of them on, you can maximize water retention, because by pulling more salt back into the body, water follows salt. So you can, if you want to have the most water retention, you're going to turn on the aldosterone to increase the sodium gradient even more, and have the aquaporins on so the water can follow it. So that's why we have both ADH and aldosterone in the renin-angiotensin mechanism, to really keep our blood volume up.
I have a question about the ADH.
Uh-huh.
You say it inserts aquaporins, or does that mean it's like activating the aquaporins, or that it's actually inserting more aquaporins into the membrane?
So I kind of talked about way in the beginning of the semester, like one of the cool things you can use endo and exocytosis for is, so like here's my cell. I might have a little vesicle that has the little aquaporins just in the membrane, but they're not actually active doing anything. They're just lying in wait. And there's some receptor, for ADH. And basically what the ADH does is going to turn on some second messenger pathway that ultimately causes this little piece of membrane to fuse and now those aquaporins are actually you dirty dog. Now those aquaporins are actually an active part of the membrane and can allow water to move across. So it's so by inserting it, I should say, it kind of starts the process of inserting them. So let's see. So any questions about any of this? There's a lot going on. I promise you're going to have to be going home and sitting with it and really kind of making sense of it all. But you are responsible. You should know what's going on in the different sections. You should understand what's going on in the collecting duct. You should have an understanding of what is the osmolarity and kind of amount of filtrate moving through the different parts of the tubule. So again, it's just going to take time sitting with it and really kind of following along. Like I mentioned, the kidneys are probably one of the most kind of involved things. Just as a little aside, just because it's kind of fun. If you look at the loops of Henle across different kinds of animals, depending on how much you have to worry about concentrating your urine, right? This loop of Henle is what gives us the gradient and allows us to concentrate our urine. how I gotta draw them better. I really do not like the new interface here at all. Think of a beaver. It's supposed to be a beaver. You know, who basically lives in water. He lives in fresh water. That's his thing. He does not have to worry about really conserving water. He's always in water. They have really short loops of Henle because they don't need to have this really intense gradient to pull the water back into their body like we do. You know, on the other hand, if you take like a little kangaroo rat, let me put him. Supposed to be in the desert with some cacti and stuff. Kangaroo rat, they literally don't have to drink. They get all the water they need just from the metabolic water from, you know, cellular respiration. But they have loops of Henle that are way longer relative to ours. They have much more of the juxtamedullary versus the cortical nephrons. They have an osmotic gradient in their kidneys that is up to, I think, around like 7,200 milliosmolar. It's somewhere like 6 times saltier in their kidneys than us. They just basically can pull back almost all the water and just have some little bit of sludge that comes out, right? So, you know, different kidneys are different. So if you're like thinking about that, I think that's interesting. That loop of Henle is what allows us to concentrate our urine. If you're a beaver, you don't really got to worry about it. If you're a kangaroo rat or something, you really got to worry about it. If you're a human, you kind of sort of have to worry about it. Okay. I'm not going to spend a lot of time, but I just want to pay a little bit of attention to the juxtaglomerular apparatus. So juxtaglomerular apparatus, it's basically this little place at the glomerulus. Juxtaglomerular just means near the glomerulus. It's specialized cells. Maybe I'll just write it out to make it easier. So where the DCT and the arterial meet, you have these specialized cells. And what they're basically doing is they are monitoring the processes happening in the nephron. So this is going to I'm just gonna monitor its blood pressure, particularly these juxtaglomerular cells and this afferent arteriole, monitoring the blood pressure. Also monitors composition of the filtrate. And basically, these are both ways to make sure that the processes in the nephron are happening properly. We've already talked about how important the blood pressure is. The blood pressure is what creates that net filtration pressure. And we want that net filtration pressure to be, in the right zone. So you are making the filtrate at that 100 mils per minute. If it's too low, you're going to not make enough filtrate, and it's going to be too slow, and things will spend too much time, too much stuff gets reabsorbed. If it's too fast, there's not enough time to reabsorb the stuff as it goes through the nephron. So you got to have the blood pressure just right to keep the glomerular filtration rate just right. You can also, by looking at the composition of the filtrate, know that something's wrong. If things are too watery or not watery enough, you know that things have been moving through too fast and haven't had a chance for proper processing. So what these will do, the main thing they'll do is if the processing is too slow, these juxtaglomerular cells, these are the ones that release renin. So if the juxtaglomerular apparatus decides either the blood pressure is coming too low to keep the filtration happening well, or it sees from looking at the composition of the filtrate that it looks like things are going too slow, these cells will release renin, and I don't have to tell you what happens then, right? You all know it's gonna then cause everything we talked about. Ultimately, create angiotensin 2, all the vasoconstriction from that, as well as ADH, as well as the volume increases because of aldosterone and ADH. This can also be activated directly by the sympathetic nervous system. I'm just saying ANS, autonomic nervous, maybe I should just say more specifically by sympathetic nervous system. So, if your brain decides we need to jack up blood pressure, it can actually trigger this. And again, if it's more of a fight or flight thing, it's actually going to constrict the afferent arterioles and kind of minimize the amount of blood going into the kidneys, because you want the blood going to your muscles and everything at the moment. So this can also be tied into things that aren't purely about kind of kidney function as well. So just kind of putting that out there. But you should know juxtaglomerular apparatus, monitoring blood pressure as well as filtrate composition, and adjusting blood pressure in particular, as well as can be constricting vessels, incoming and outgoing vessels, to try to make sure that the glomerular filtration rate is staying in the target zone. So there's that.